Hello all, it's Ferdinand tutorial. Let's continue from where we stop on amino acids. Now in our last class we are talking about the 20 amino acids, the structures and how we group them. Today we'll continue with the characteristics of amino acids. Now when we are talking about characteristics of amino acids are classified based on two characteristics. One, we have the physical characteristics or properties, and we have the chemical characteristics or properties. Now when we are talking about physical properties, we said that physical properties are those properties that we can see that amino acid possess. Now, example of such physical properties, they are 5 in number. Now, they include optical properties, they include solubility, they include high melting points. Amino acid also have isoelectric properties and absorption spectra. Now, let's start with absorption spectra. Now, amino acids, like let's say they are aromatic amino acids, where we have the phenylalanine, we have histidine, we have tryptophan. Now, if you see this amino acid, they have a very high absorption spectra. Now, they has a radioactive ray up to 270 nanometer. Now, why do they have a high absorption spectra? That's the question we should ask. Amino acid have a high absorption spectra. One, because of its aromatic structure. Now, if you look at these 4 amino acids, where we have histidine, we have tryptophan, where we have tyrosine and phenylalanine. Now, these 4 amino acids out of the 20 amino acids that are the monomeric units of protein, these 4 amino acids has a very high absorption spectra. Why? Because they have an aromatic side chain. Are you getting it? Now the structure have a very large structure. Are you getting it? Making it to have a aromatic side chain. Now the next property that we'll be looking at is what? High melting point. Now amino acids have a very high melting point. Why? Now they have a very high melting point because of its, let's say, it has lattice energy joining the amino acid. Are you getting it? Making amino acid, before it melts, it takes a very high energy at about 200 degrees Celsius. Now having known that, let's go to another properties. Another property is solubility. Now when we're talking about solubility, amino acid dissolves in polar solvent like water, or do not dissolve in non-polar solvent like benzene, chloroform, and ether. So now the next property there is that isoelectric property. Now when we're talking about isoelectric property, remember we said that isoelectric property is a property exhibited by amino acid when it has no neutral charge. It has no net charge, sorry. It has no net charge. That means that amino acid has a neutral charge. Are you getting it? Now at this point, amino acid has a neutral charge. That's the point where we call what? Isoelectric property. Now what makes amino acid has a net charge? Now remember in our last class I was saying something. I said that if we have this as COO minus, now we have here as NH3 plus, now we have here as H, and we have here as the arrow group. Now I explained something in our last class. I said that here now this is a carboxylic functional group. This is what? An amino functional group. An amino functional group. Now you see these signs, are you getting it? These signs, they are balanced. Are you getting it? Making this amino acid to be what? To be neutral. Are you getting it? That's now at this point where amino acid has no net charge. That's what we call what? Isoelectric pH. Are you getting it? So amino acid exhibits what? Isoelectric property. Now the next one we'll be talking about is where we have the optical properties. Now when we're talking about optical properties, now remember that amino acid have a chiral carbon atom. Are you getting it? Now when we're talking about chiral carbon atom, I say that chiral carbon atom is that carbon atom that in which there are 4 groups that are joined to the carbon atom, 4 different groups that are joined to the carbon atom. Now that carbon atom, we call it what? Chiral carbon atom. Now out of the 20 amino acid, the 20 amino acid exhibit, that's 19 out of 20. The one that do not exhibit is what? Glycine. Are you getting it? So it doesn't have a chiral carbon atom. Now from the second amino acid, that's from alanine down to the 20th amino acid, exhibits chiral carbon atom. That is to say that they have the D configuration and the what? L configuration. Are you getting it? Why? That is the dextro configuration and the what? Levo configuration. That is it for the what? The physical properties of what? Amino acid. Now let's go back to the chemical properties of amino acid. Now we are talking about chemical properties of amino acid. Now amino acid is amphoteric in nature. That means it has basic property and what? An acidic property. Basic property in the sense that it accepts electron, and the acidic property in the sense that it gives out what? Electron. Now, a compound can as well act as basic and as well act as what? As acidic. Amino acid does that. Now, there are 4 typical reactions in amino acid. Now, the 4 typical reactions include, now let's write them down. We have the first one, decarboxylation. We have the second one, transamination. We have the third one, non-oxidative deamination. And the fourth one, oxidative deamination. Okay, this is the 4 typical chemical reaction that an amino acid undergo. Now let's look at this first one. Now remember, here is decarboxylation. Now when we are talking about decarboxylation, we say that decarboxylation is the removal of one or more carboxylic functional group from a compound. Now, the decarboxylation of amino acid leads to the biogenetic primary amines. Are you getting it? Leads to biogenic primary amines and what? Carbon 4 oxide. Now, if you look at histidine, now remember in histidine, histamine was formed. Are you getting what I'm saying? Now, the formation of histamine from histidine is as a result of what? Decarboxylation reaction. We know that histamine is a hormone that is produced in the body tissue that is responsible for hypersensitivity. It's one of the hormones that is responsible for what? Hypersensitivity in the body. Now, let's go back to transamination. Now, when we are talking about transamination, we say that transamination is the transfer of amino group from what? Amino acids. Now, when we are talking about transfer of amino group, let's say we have an amino acid and what? An alpha keto acid. Now, if we have this amino acid, an amino acid plus an alpha keto acid. Now, when they react together now, when we are talking about transamination, when they react together, now this amino acid, the amino group is being transferred to the keto acid, and this keto acid is being transferred to the amino acid. That is, if this one is 1, this one is 2. Now, there is an amino group here. Now, this amino group will be transferred to this keto acid. Are you getting it? Are you getting? Forming a new amino acid here. And this, the functional group here will also transfer here, forming keto acid. That means 1 will become 2 and 2 will become what? 1. That is what we call what? Transamination. And the enzymes that is responsible, the enzyme that is responsible here is what? Aminotransferase. Are you getting? Aminotransferase or transferases. Now we have non-oxidative deamination. It involves the removal of water from certain amino acids. That's for non-oxidative what? Deamination. Now it also produces a product, like it has a final product which contain alpha keto acid and what? And ammonia. Now when we come here, oxidative deamination, here water is needed. Are you getting it? Unlike deamination also, it also produces what? Alpha keto acid and what? And ammonia. Now apart from these 4 typical reactions, there are other reactions like neutralization reaction and what? And esterification. Now when we are talking about neutralization reaction, we say that neutralization reaction, it involves when an acid reacts to the base to form what? Salt and water. Someone might be asking, what are we using amino acid as? Is it an acid or as a base? Now you can use it anyone that suits your reaction. Now when you use it as an acid, it also forms salt and water. When you use it as a base, it also forms salt and water due to its what? Amphoteric nature. Now, having known that, let's talk about amino acid modifications or derivations. Now, when amino acid, remember that we said amino acid are monomeric units of what? Of protein. That means that amino acid can be modified after we have used it to form peptide bonds or what? Proteins. Example, amino acid that are found in lysine, prurine. Are you getting it? Now, these amino acids that are found in lysine and prurine, let's take for instance 4-methyl lysine, 4-hydroxyl prurine. Are you getting? Or 4-hydroxyl lysine. I don't know if you are getting what I'm saying. Now, if we use this, let's take for instance, when we are talking about lysine, let's modify lysine. If we have lysine here, remember we have C here, we have COO minus, we have NH3 plus, we have H here, and this is the arrow group. And here we have CH2, 4, and we have what? NH3. Are you getting it? Now, if you want to modify this amino acid, you know the meaning of this 4 here now. It has CH2 into 4 places. That is it. Now, if you want to modify this amino acid completely, now when we modify, we will have 4-hydroxyl lysine. Are you getting it? Let me modify it. If I have here, I have C here, I have COO minus, I have H here, I have my amine group here, NH3 plus. I also have now, let me open it, CH2, I'll open it, CH2, I'll open it, CH2. Now what do we call it for? Now 1, 2, 3, 4 hydroxyl. Are you getting it? 4 hydroxyl what? Lysine. So this is the modification. Are you getting it? This is the what? This is the modification of what? Of lysine. That is it. Now, having known that, now let's talk about some functions of amino acid. Now we know that we can say that amino acid has the main function of amino acid is to form the monomeric unit of what? Protein biosynthesis. Not only that, there are some amino acid, even those that doesn't occur in protein, that have functions. Now, example of them, we have glycine. Although glycine occurs in protein, it's a monomeric unit of protein. We have glycine, we have ornithine, we have citrulline, we have gamma amino butyric acid, the one we call GABA. Are you getting it? The one we call what? GABA, and other amino acid. Now, ornithine here, we say that ornithine is used for what? Urea biosynthesis. Also, citrulline is what? Also used for what? Urea biosynthesis. Gamma amino butyric acid is is an amino acid that acts as what? As neurotransmitter. Now, we're talking about glycine. Glycine serves as is an amino acid that serve as conjugation or detoxification what? Reaction. It is present because conjugation or detoxification reaction. So that is all for this class. That is how amino acid class ends. So we'll be starting again in our next class. So I urge you to share to your friends who don't know about Ferdinand tutorial. Tell them about what we do here. Comment and subscribe to this channel. Also subscribe to this channel. We'd love to see you again in our next class. Thank you.